Hello, my name is Martim Pardal and I'm a physics student at Instituto Superior Técnico in Lisbon, and I would like to present my master's project. From very early, we learned about cells as building blocks for all life on Earth. We know that cells are complex systems with different working parts, and that cells and unicellular organisms alike are able to divide, to grow and multiply in a process where the shape of the organism is constantly being changed and adapted. We all started as a single cell that quite extraordinarily is able to produce a full, complex organism when it increasingly divides itself. Those resulting cells will have different functions and will change their shape accordingly during development. So why do organisms of all kinds have the shape that they have, and what determines it? The biological process of development of form and shape is called morphogenesis. One example of a morphogenic process is the initial step of gastrulation in the embryonic growth, where there is a spatial reorganization of cells and a breaking of symmetry that's known as invagination. This is also when the first differentiation of cells occurs in the embryo, showing the connection between morphogenesis and cell differentiation. The study of the mechanisms behind morphogenesis has been and continues to be a pressing subject. It was Alan Turing that, inspired by patterns found in nature, that first predicted a model for the mechanism. He proposed pattern formation as being a result from the diffusion of two chemical substances on a surface, one with an activating function and the other working as an inhibitor. He labeled these chemical substances as morphogens. So morphogens are segregated and diffused through tissue, and cells respond according to the local concentration. To describe this model, we use the reaction diffusion partial differential equation, which shows similarities to the standard heat equation. This class of equations was used by Turing to describe pattern formation, and were the basis of the work of Cummings on the influence of morphogens on the changing of shape in organisms. In this approach, morphogens evolving along the surface of cells may change surface tension, inducing a change of local curvature that results in a change of shape. On the other hand, if we consider the free energy of a surface, the most favorable configuration of the system will be the one that minimizes it. So, a functional that considers the physical constraints, in particular the elasticity of the membrane, can be proposed, and by minimizing it, we obtain a shape equation that will give us the most favorable configuration for a cell or multicellular aggregate. This is what Elfrich proposed, and the general shape energy equation has proven successful in recreating the characteristic disc shape of red blood cells. Further developments have been recently obtained when Mikuki and Zhou, extending on Elfrich's functional, were able to relate protein diffusion on the membrane as a function of the curvature of the membrane. The consideration of these molecules provides a more biologically realistic model. What our work proposes is using these approaches as a starting point to produce a model describing the replication mechanism of a cell by mitosis and eventually the invagination process. Hopefully, this can lead to a better understanding of the mechanisms behind morphogenesis. Thank you. Hello. My name is Sofia. Não é este. Não é este. Estou com dúvida.